Hello and welcome to the Imaging Wire Show. My name is Brian Casey. I'm managing editor of the Imaging Wire. Got a great episode for you today. We'll be talking about collaboration in the radiology AI sector, specifically ScreenPoint Medical's recent acquisition of Biomedic. And our guests are Professor Nico Karssemeijer. He is Chief Science Officer of ScreenPoint, and Professor Mats Nielsen. He is CEO of Biomedic. Gentlemen, thanks for being with us today.
Okay, thanks for giving us the opportunity, Brian.
Of course, our pleasure. So let's start with introductions. Nico, can you go first? Tell us about yourself and about what ScreenPoint does.
Yeah, I'm a founder of ScreenPoint Medical, and we founded the company about 10 years ago, starting out to help radiologists with reading screen mammograms and gradually expanding with the company. It's been an exciting time for us. I have a technical background, but worked for almost my entire career here at Radboud University in the Netherlands, in Nijmegen. Within the radiology department, we developed many applications, and it's been exciting to take one of these into the field as we are doing now with Transpara, with the company.
Very good. And Transpara has been the focus of some really exciting papers recently that we'll be talking about a little bit later. Mats, tell us about yourself and about what Biomedic does.
Yeah, thank you, Brian. So I'm have been for many years professor at University of Copenhagen, working in AI and the applications in medicine in various areas. And then 15 years ago, we agreed on that in order to really get it to work with patients, we needed to do it in a company setting also. And we have been working in Biomedic primarily with having biomarkers for use in clinical trials, but also came to the use of those directly with patients. And that's where we are today, and that's why we are here. We have spun out a couple of other companies that are in the startup phase of medical imaging, cerebroprenatal, working on brain imaging and fetal ultrasound.
All right, very good. So, Nico, in early January, ScreenPoint announced that it was acquiring Biomedic. Can you tell us a little bit about the acquisition?
Yeah, it was a fairly straightforward process. It was following up on a very long standing collaboration. I think we talk about that later a little bit more. But so for me, it's been a logical step for quite a while, especially since Biomedic spun out some of their other activities. And I think Mats and I were pretty much aligned on it. And I think gradually, since we seen so much activity in personalized screening, and I think everyone in the board of the company, the shareholders, came to realize that actually risk is a very logical next step to move on to within ScreenPoint, and then that made the acquisition of Biomedic very logical and also very attractive.
So ScreenPoint makes AI for analyzing mammograms, and some of the studies that have been used have found that it's been really useful in figuring out if a mammogram is normal or suspicious, triaging them. Biomedic makes kind of risk analysis software to figure out when a woman comes in, is she at higher risk for breast cancer? Nico, was that what attracted you to Biomedic? Because this kind of union of what ScreenPoint does in mammography AI and risk analysis?
Yeah, absolutely. So we are in the business with Transpara. It's the product that we are now selling worldwide. It's validated in many studies. It's very successful. It's helping the radiologist to interpret the mammogram. But we found that even if a radiologist says, well, it's negative or benign, that actually the analysis that we do in the mammogram is still quite predictive for future breast cancer. So that's even without Biomedic's technology, but that's just analyzing areas in the mammogram which the radiologist probably also observes and sees, and he thinks, well, it's not actionable. It's not enough reason for me to work up a finding, to do a recall. But still, they still are risk factors and pretty predictive. And so that is where we are coming from. But with Biomedic, we've been working Biomedic, and in our collaboration also we've been working on. It's also something in the breast tissue in general, not really a finding at a certain location. That is an area that may already be a developing breast cancer, but just signs in the tissue that indicate that there is a higher risk of developing breast cancer. So, and we are merging these two components in what we are doing. And maybe Matt, you can tell a little bit more about that.
Matt, can you let us know like how exactly Biomedic's technology works?
Yeah, so basically we started out from the total opposite viewpoint, that we developed the technology for use in clinical trials, where as a safety marker, you want to survey that the women are not getting an increased risk of breast cancer. But it takes very many years to figure out whether they were in increased risk of breast cancer. So we wanted to develop markers that could, on the short term, by just looking in the mammogram, look at the risk of future breast cancer. So that is not looking for the early signs of cancer, but it is just looking at how is the tissue structured in the breast. So it's well known that breast density is a risk factor. So that's the amount of fibroglandular tissue in the breast. But the whole structure, how it's connected, how it's distributed, also carries signs of the risk of future susceptibility to have cancer growing in the breast. So that's what we decided to use markers on. And this was actually just in the verge of the deep learning area of medical image analysis, but using machine learning to look at a large collection of images. And actually our first study we did together with Nico Karssemeijer on Dutch images, where some of the women would get cancer later on, and the other part of the women, they were cancer-free for the same period. And then simply having the machine learning to try to figure out what is the difference in distribution, texture, the visual features of the mammograms. And that has been quite a successful approach, and has many more signs of potential future cancer than what just looking at the density in the breast has. So you can say it's kind of a density on testosterone that we are using here.
So with your technology, just to clarify again, with your technology, it can tell if a woman may be at higher risk for cancer in the years to come, even if there's no cancer in that mammogram right away. Is that correct?
Yeah, that is how it is. So basically, whether the tissue structure is so that the risk is higher, either. So you can one way I visualize it mentally is also saying, does she have more votes in the lottery? Now it's just a prize you don't want to win, but that's how I look at it.
Now, Mads, you mentioned that this is being used in research. Has Biomedic commercialized this technology yet, or is that something that's kind of still in development?
So we have only developed it and used it in studies, also in commercial studies, but not for clinical usage yet. So that's what we are looking forward to, and that's also why, seen from my point of view, this acquisition was so obvious, because ScreenPoint is market leader in breast cancer detection, and then getting our technology out, helping that in stratifying breast cancer screening in the best possible way seems a logical step.
Yeah, definitely. Well, it seems like, Nico, combining risk analysis of mammograms with cancer detection is going to be a really powerful combination. Can you talk about how the biometric technology will work with Transpera in a screening environment?
Yes, absolutely. So this is first of all, I think what we are after is improving the screening process. So although biometrics technology is predicting also the risk of cancers that are not even detectable, we've developed a product at the moment more with the aim of finding the cancers that are already detectable. And so there's the complementarity of the two approaches is so important. So because we see that with new technologies that are nowadays more and more accepted in clinical practice, such as breast MRI, contrast-enhanced mammography, and to a lesser extent, whole breast ultrasound, but especially the very sensitive techniques such as contrast-enhanced MR. We have seen with the DENSE trial here in the Netherlands, but also in other big trials that there are many more cancers that are already detectable at the stage where women come for their mammogram, but they're negative. They're not detectable on the mammogram yet, either because of density, they're just too small. But you do an MR in the group that just had the message that their mammogram was okay, there's no cancer found. If you do an MR on those women, we've seen with these trials that you find another 20 cancers on top of the 6 to 7 you find with mammography. And those are the cancers that we want to find with our tools. But those trials are typically done with a selection of women based on their breast density or using a risk model based on clinical factors. But we've seen that these are not so easy to use in practice, and in particular, density is actually not very effective to predict breast cancer. The tools we are developing now are much more powerful. That means that will be able, with as soon as we have developed this into a product, to provide a tool to radiologists to very easily and quickly run an analysis on the mammogram, and then when the women are still in their in the center for their screening, they can immediately tell, okay, you might consider do a supplemental exam because you're high risk, and that's going to be a much more effective approach than the current clinical practice of using clinical risk models, which requires self-reported risk factors that may be unreliable or not known. And so we are testing at the moment our risk prediction tools and comparing them to existing models based on clinical risk factors, and we find that they're much more effective.
Interesting, because this idea of basing screening around risk is relatively new, because for many years, determining who got mammography screening, what women got mammography screening, was just based on age. And now we're starting to have an understanding that that's kind of a one-size-fits-all approach, that maybe you might be screening some women too much and you might be screening other women not enough. Do you see risk being used to guide all screening at some point in the future? And if you do, how might biomedics technology integrated with Transpera kind of help inform that?
And I think in Europe, programs are thinking about maybe to also adjust maybe the frequency of screening. That is something we are not immediately considering. We are really focusing on who should we recommend these supplemental screening exam, because that is proven now with randomized controlled trial to be very effective. It's implemented in some countries, but then with the selection mechanism based on density, which is not so effective. And that's where we see at the moment the biggest opportunity. There's a lot of resistance in using these risk tools also to adjust frequency of screening, although some think about it. This is not really new. in the scientific community, personalized screening based on these models been discussed for a long time. But what we see now is the time is getting right, because we have the evidence now from several trials, like the DENSE trial, and also the BRAID trial recently in the UK, that these new screening methodologies are accepted by women with breast MR and highly effective. So, and also cost effective. So now all this data is there. We can think about starting an implementation in clinical practice, and then we want to provide the tools to make this even more effective.
Yeah, well, it sounds like it really has the potential to kind of change the equation around breast screening, which is kind of hard to believe, but always been a little bit controversial, at least in some circles. Mads, so this is all really exciting. How much more technical integration needs to be done to bring the biomedic technology together with ScreenPoint's Transpara?
So in the lab, we have already integrated the products, and we have also tested them together to see that the localized findings that Transpara is scoring and the overall tissue scoring that they actually add up. So they have complementary information, and we have one publication out in radiology about how the risk segregation gets even higher by combining the two approaches. So in the lab, things are done, but in the real world, we are now starting the full software development process and so on. So we, over the next months, can come out with a software product that can be approved by the authorities.
Right, very good. We'll look forward to seeing that. So, Nico, if we can maybe step up a level and talk a little bit about the broader market for AI for breast screening. I mean, there have been so many really exciting papers published in the last year on the use of AI for breast screening. Many of them did use the Transpara algorithm. Do you have any thoughts about kind of where we're going with this right now? I mean, it seems like in Europe, at least, we might be on the verge of being able to use AI instead of a double reader in some programs. Do you think that's going to happen?
Well, it's actually already happening. I think we are very successful with that in particular in Denmark, also in Sweden. In Denmark, that has been adopted for the larger part of the country as the way to go. And I think maybe I should formulate it a little bit more precise that you did. So we are not replacing a second reader, because in our approach, we think there's actually a lot of value to have a second reader. We've also seen that in the prospective studies that we did, but it's not necessary to have a second reader for every mammogram. So what we are doing is we're removing the bulk from the second reading process based on the Transpara outcome. So that's the triaging. But at the same time, the radiologist, we provide them with the output of the software during the reading process, so that they can use this information to become better. That holds for both the readers that still do the double reading for the mammograms with the highest risk of breast cancer, and that varies. Like in Copenhagen, that's about 25%. In the Masai trial in Copenhagen by Christina Lang, it's about the upper 10% is still double read, but the rest is single read. So you have a massive reduction of the workload, and that was initially actually the incentive. That's why they wanted to implement AI, because of the workload issues that they were facing and the backlogs in the screening. But what they found, a little bit to their surprise, maybe to some of them, that the cancer detection rate increased also very strongly, due to the effect that for the majority of the mammograms you actually have a third opinion from the AI system, which is really good, and it's outperforming these days for 2D mammograms most radiologists. So it's really for the screening mammograms, I should say. But that is that leads to a big improvement, and also the radiologists enjoy working with it. I believe they also get better with it. And we've seen now the latest results of the Masai trial, which already presented at ISOBI. They will be published in the Lancet Digital Health next month. They actually show an almost 30% increase in cancer detection with a reduction of workload. That is fantastic news, of course, for the screening programs in Europe that can solve workload issues, but at the same time become better.
That's a great point. So you're kind of having your cake and eating it too. You've got less workload, but you're detecting more cancer, and that's just an amazing combination. Mats, what kind of future developments do you see in integrating the technologies between ScreenPoint and Biomedic? Like, where do we see this going in the future?
Yeah, so I think the steps here by really improving the risk of future cancer estimation in the screening program for the secondary use of imaging, and having really trials on that, showing the value there in implementation in the clinics and large scale implementation. That's what we are looking forward to, showing that the it's not just better risk assessment, but it also has clinical impact. That's that's personally what drives me in all this, to see this clinical impact. Then, of course, technology can always become better and integrate more information. So next steps in the future may be to take more clinical information of the women into account from the electronic healthcare records or other readily available information to make the risk segregation, the risk estimation even finer than what it is today, so that we can, in the way Nico explained, drive the resources in the directions of where they are used best. Because we cannot and should not offer supplementary imaging to every woman, but we should do it to the right women, and that's what we are aiming at.
All right, very good. Nico, the European Congress of Radiology is coming up later this month. Any important papers that using the ScreenPoint technology that we should kind of keep an eye out for?
Yes, of course. So I think it's for us, it's going to be we've never had so many presentations scheduled as this year for ECR with our technology. So we're not presenting ourselves, but it's our clinical partners. Very important. Hopefully, we'll get some late breaking news also. One from Copenhagen, where they have implemented our solution in the regular screening program. So not a risk product, but Transpara. They've been evaluating this ever since, and some publications have come out already, also showing a strong increase in cancer detection rate with a reduced workload. But what everyone is waiting for now is, also for the Masai trial, is does it have an effect on the interval cancers? Does AI, it has a higher cancer detection rate, but does it find the right cancers earlier, especially the interval cancers, is a very important question. And we hope to have some news on that on the ECR. That would be fabulous. But we have also presentations from the trial in Spain, in Cordoba, where they are testing autonomous AI, also with tomosynthesis. That's actually something we didn't touch upon yet for the risk, but of course we're focusing very heavily, of course, on the US market with a risk product, because their implementation seems to go much faster than in Europe. And that's all Tomo, and the risk product that we are developing will also be multi-vendor, just as Transpara and for tomosynthesis.
And when you say autonomous AI, that's basically running the AI without any radiologist involvement in that, correct?
Yeah, that's fascinating. That's where the field may be going in the future. I expect it will, because at some point, it's pointless that radiologists read large volumes of mammograms or tomosynthesis studies while there's nothing more that to be found, because AI is getting such a high predictive value that what's coming out there.
All right, very good. So Mats, another project that you and Biomedic are working on is the Eurostar project. Can you tell us a little bit about that?
Yeah, so we have been working together with the ScreenPoint in several European projects. One is Eurostars project. Another project is the Azure project, and especially in the Eurostars project, the Copenhagen Screening Program was involved in that project also, and that's where we originally saw the benefit of combining the two approaches of the localized finding from Transpara and the more systemic risk factors coming from the biometric technology. So that's actually a European project that has led a couple of years later now to the merger of the two companies, because the technologies are so that we could see that they supplement each other so much that it's better to put together.
Well, some great discussion today. I'd like to thank Professor Nico Karssemeijer of ScreenPoint and Professor Mats Nielsen of Biomedic for being with us. Gentlemen, thanks so much. We appreciate your time.
It's been a pleasure.
All right, signing off for the Imaging Wire. My name is Brian Casey.